just about ready to go now. Aiden, I hope you're recording, so we're good to go. All right, fantastic. And I presume that Richard is as well, though he does it remotely from time to time, so he may not be listening to us at the moment. Okay, great. So before I even get going, I put up this just to remind people that Aspirational Neuroscience is hosting a satellite event at the annual meeting of the Society for Neuroscience. It's in San Diego this year. So for anyone who's heading out that way to participate in the annual meeting, please come check us out Monday, November 17th, 7 to 10. That's when Ken is going to be handing out the Aspirational Neuroscience prizes, right? I mean, if you think that's going to come together, I think it will. Yeah. And we look forward to especially the conversation that will be happening there. We're planning to have a panel with several of the leading scientists who would be of relevance for memory decoding, because of course memory decoding from a static set of brain data is the challenge this year. And yeah, it'd just be great to have you guys there. So that's a quick reminder. Now we might as well move on. Let me see if that works correctly. There we go. All right, so this is today's paper, Connectomic Traces of Hebbian Plasticity in the Entorhinal-Hippocampal System by Grasso, Corteze, and Schmidt. And I was particularly interested in this paper because it's an example of work that you can do by relying purely on the data that's inferred from volume electron microscopy. And you'll see in a moment how that is when I get into the methods. So they were looking into hippocampus and entorhinal cortex. And now, just as context, we've had a few previous journal club discussions about, recently, about evidence for non-Hebbian forms of plasticity. But the default model is that LTP at synapses in the hippocampus creates associations. And then eventually those associations, those changes are consolidated into stable memory engrams. And so learning changes synaptic weights, either by strengthening them or by weakening them, or even by pruning, i.e. abolishing those connections. So based on this, going into it with this default model, the authors listed a set of questions, important questions, such as what fraction of the synapses in CA1 and CA3 and other areas are at any given point in time in a state that actually results from learned LTP configurations. In other words, which ones are actually involved in the learning, in the storage of new information, as opposed to relevant for other reasons. Is that storage graded or is it binary? And if you want to, you can look back at one of our previous journal clubs, which was about the paper by Dorkenwald et al. From 2022. And we presented that study, which is about mouse primary visual cortex back in July, which was an example of looking into the possibility that you have binary states there, or at least that's kind of what came out of it. And then another question is, are there specific hotspots of synaptic imprinting? You can look at Sievers et al. From 2024 there. That paper, which is called Connectomic Reconstruction of a Cortical Column, has actually also been on our radar for the journal club for a while, but I don't believe it's been presented yet. Then another question is the storage primarily in the local circuit, or does it involve long-range connections? And for instance, look at Utiepo et al. In 2025. And that paper, called Synaptic Architecture of a Memory Engram, has also been on our shortlist for a journal club presentation, but I also don't think we've done this one yet. And then finally, how is the medial entorhinal cortex involved? So in this paper, what the authors do is they carry out a systematic analysis. They're mapping the apparent weight distributions of the connectome, and they have a particular way of figuring out the apparent weight distributions. And they're looking at the relation of that to Hebbian preconditions. So there's some relationship between same-axon, same-dendrite, SASD, paired synaptic configurations, and they analyzed 5.85 million synapses and tens of thousands of paired synaptic configurations for that. We'll get into the paired synaptic configurations in just a moment. They used 3D EM-based automatic circuit reconstructions from hippocampal area CA3, CA1, also layers 2 and 3 of the medial entorhinal cortex. These all belong to that same hippocampal-entorhinal system that is so crucially involved in episodic memory. And they found systematic and unique synaptic weight distributions. So for example, they found that almost 50% of the synaptic weights in CA1 are in a Hebbian-consistent state, but that's not 100%, that's just 50%, right? So this is going to become important, and particularly in the molecular layer of CA1. And they found that CA3 uniquely exhibited small synaptic weights with indications of learned states. So they seemed to be small not just because they hadn't become large yet, or because they were not involved in learning, but they seem to have learned a small weight specifically, or at least that's kind of what rolls out of the interpretations there. And that's particularly in the lower layers of CA3. And in the medial entorhinal cortex, they found only about 20% of learned synaptic configurations if you interpret learned as being related to these paired synapses. Okay, now about the methods. So importantly, there is no stimulation or learning paradigm in this study. The brains are from 4-week-old mice that were analyzed using state-of-the-art connectomics methods. They had 9 subvolumes in CA3, and this, as Ken already pointed out at the beginning of this session, is from a data set that had already been published, Salmons, Vizier et al. And each of these 9 subvolumes was either a volume that was 175 by 100 by 57 micrometers or 100 by 100 by 50 micrometers. So they had a few different options there, but these are the same options they used in the volume size in other areas. So for example, in CA1, they used 3 subvolumes from Corteze, Grasso et al. And each of those are 100 by 100 by 50. In the medial entorhinal cortex, in layers 2 and 3, they used one subvolume each from previously unpublished data. So I guess we can't refer back to anything that has been found in that area yet. Each of those 175 by 100 by 57 micrometers. They aligned those and segmented them using voxeletics, resulting in a volume object in volume objects that had probabilities of belonging to either axon, dendrite, or other. They were able to find axon spine synapse probabilities and the size of the synaptic interface. For reference, voxeletics is a popular neuroscience AI platform by Scalable Minds that's used to process volume EM data. And it's able to do this sort of thing, to automatically align data sets, segment neurons, detect synapses, and generate complete connectome matrices. So then they carried out same-axon, same-dendrite paired analysis, and this was done as in Mata, Burning et al. 2019. So you can read more about it in that paper. That's the paper entitled Dense Connectomic Reconstruction in Layer 4 of the Somatosensory Cortex. It's probably also a familiar paper for some of you. And they looked at the synapse sizes for these SASD, same-axon, same-dendrite pairs, where the synapse size is considered related to the synapse strength. There's a lot of evidence that there is a relationship there, a strong relationship. And they computed the average for each pair, and they also computed the coefficient of variation within each pair. And then they assumed or interpreted this as variation being an indicator for consistency, where 0 means they're completely matched. And for the non-SASD pairs, what they did is they picked random synapses, they shuffled that. They studied the SASD paired connections in comparison to the random pairs of synapses for possible relation to the history of Hebbian plasticity. So this, I found that to be actually a very nice way of doing things. All right, let's get into the results then. So this is figure 1. about the analysis of synaptic size distributions. And I added just a little bit of information in each to make the figure more readable. So they had found 1.6 million synaptic spines in the the CA1 subvolumes, the 3-volume subvolumes they used there. They found about 3.4 million in the 9 CA3 subvolumes. So, I mean, since those are 3 volumes and those are 9 volumes, you can understand why the difference in the number of synapses. They found 275,000 synapses in layer 2 of EC and 464,000 in layer 3. And just as a reminder for everyone, sorry, CA3 is receiving a lot of input from entorhinal cortex. CA1 receives input from CA3. It also receives input from entorhinal cortex, which I forgot to add here. And then the layers of L3 and L2, those are, of course, the hippocampal input layers. What you see in A is the circuit sketch, which is giving you some idea of how everything is connected. Notice that entorhinal cortex layer 3 mainly leads to CA1 and the subiculum, but the subiculum is not shown here. And entorhinal layer 2 is the input to CA3 and the dentate gyrus. Then if we look at figures 1B to E, so all of these, those are the synaptic size distributions for all of the spine synapses in each of the regions studied. You notice that the x-axis is logarithmic. So this is, I mean, you could see that, of course, over 0 and 1, those are the largest synapses, and then down there is the smallest synapses. And notice the bimodal appearance. And if you've been to our previous journal clubs, you're going to recognize this. We've seen this before. You see this shoulder on here. It's giving you that bimodal appearance that you can't really fit a typical logarithmic shape to the distribution. The medial entorhinal cortex data in other over here, so L3 and L2, doesn't look like that. It looks very similar to other data from the neocortex, like, for example, from Mora Burning et al. In 2019, with a log normal shape that's slanted towards larger sizes. And in CA1, we see large and small synapses, and they're not quite, but nearly equally prevalent. But in CA3, there's a huge difference. Small synapses are far more prevalent with a 9-to-1 ratio, but still with this clear bimodal shoulder. Sorry, I'm going to take a quick sip here. Okay, and in 1F, this is just them putting them all on top of one another. You can see the bimodal distributions in CA3 and CA1. They match, at least for the large synapses. Oh, sorry, the small synapses. And they differ, obviously, from the main sizes that you see in entorhinal cortex, where the peak is quite elsewhere. So this left a few questions. So were the overabundant small synapses in CA3 just randomly established? Or could systematic patterns of innervation be found in there? And is there an overabundance of paired synaptic configurations which could be consistent with a history of Hebbian plasticity? And then what fraction of the synapses in CA1 and CA3 would be consistent with a Hebbian state? And finally, does that depend on the layer location within the brain areas? In other words, areas where different kinds of connections are happening for different reasons in terms of what the circuit is doing there. Let me just quickly check if anyone has a hand up, because I can't see that at the same time. No, looks okay. All right. On to figure 2 of the results, the analysis of paired synaptic configurations. So here they're analyzing the paired synaptic configurations in those same-axon, same-dendrite connections in order to detect possible traces of Hebbian plasticity. And in 2A here, we just have some examples of dendrites and axons in each region, where the dendrites are these long pieces with the spines, obviously, and then axons are the little blue parts. And you can see how they form these paired connections, different kinds of paired connections. Notice here in layer 3, they're actually using some connections that are on different branches of the axon. And they form these synaptic innervations that that we're most interested in. So now in 2B, we're looking at the average synaptic sizes and their relative frequency. So we've got relative frequency up here, the average size, so the average within the pair. And again, of course, this is a logarithmic scale. The black distribution, these are the SASD, so same axon, same dendrite pairs, and the gray is randomly paired. And you can see, of course, the bimodal distribution that we already remarked upon. And you can see that clearly in CA1 and CA3, CA1, CA3. And you can see that there's also a shift to larger synapses for those SASD pairs, especially in entorhinal cortex layer 2. So if we look here, there's a shift for the SASDs towards larger synapses. And this is all consistent with a view of synaptic pairs being formed to further strengthen the synaptic connections beyond the weight that a single synapse could achieve. So if you're thinking of saturation with LTP, largest possible strength or weight, if you want to get further, if you want to get beyond that, then, well, you can add another synapse. And this is substantial in entorhinal cortex layer 2, but the effect of that is actually pretty modest in the other layers. Now in 2C, they're comparing the size variance between or within the SASD versus random pairs. Note that there's a shift to low variance pairs for SASD, so the black line. No, they switched that here, the blue line, especially in hippocampus. But even in CA1, if you look carefully, a key site of synaptic plasticity in the mammalian brain, there's really only about half of the synaptic configurations that showed an overabundance of low variability synaptic pairs. And that would be inconsistent with a view of CA1 as completely governed by saturated LTP, or at least not in this non-stimulated animal. Let me just check who it is. Hey, Ken, go ahead.
I was waiting for you to say something like that.
To trip me up.
How is this? Okay, so this paper is talking about consistency with Hebbian plasticity and saying that a certain percentage in each of these regions is consistent with Hebbian plasticity. Can you unpack that? I find that a little bit confusing.
Unpack it. Okay, so we've got two components to this, or actually three even. We've got the low variance. So low variance between two synapses in a pair. So that's assuming that they've both experienced the same history of presynaptic and postsynaptic activity. Gives you similarity.
Yeah, so
no strong variance there.
So the blue curve in these, I'm looking at C for CA3, let's say, and down near X is equal to 0, that means that they're the same size synapses, right? And there's many more of the same size synapse than
within a pair,
than would be for random.
Right.
Okay. So that is the key piece of information that is consistent with Hebbian, but I'm thinking of the Dorkenwald paper. The Dorkenwald paper definitely would not say these are inconsistent. The ones that don't do this are inconsistent with Hebbian plasticity. They would just say there's a random variable that's attached, and that random variable has gone above a certain amount.
Yeah. No, you're right. There is definitely, you don't necessarily have to interpret it that way. They're leaning pretty heavily into the interpretation where strong Hebbian learning leads to saturated synapses or saturated LTP, and that may not always be the case, right?
In any case, you don't have to slow down and discuss this now, but just as put a point in that I read the Mata paper. The Mata paper goes through this in much more depth and yet I was still confused out of the Mata paper about them saying literally, because that is, I mean, they're saying this, and this is a very, very, very strong statement, that they're finding 50% of the synapses incompatible with the Hebbian hypothesis. And
I would say with their Hebbian hypothesis.
Oh,
Yeah.
okay, so that's what I want unpacked at some point. Go ahead,
Yeah.
though.
I mean, I think it's not crazy if you think about how many synapses you also need to do other things besides storing pattern engrams, right? So you can see why, at least you would expect not every synapse to be directly involved in learning engrams. But saying, okay, so all the synapses that we see that have low variance paired, I don't know, strong or even weak, as in CA3, as you'll see in a moment, strengths, that is an evidence of Hebbian LTP, but others are not. That, I think, that's perhaps a little bit too much. So I think all they're doing is saying these are the things that is the best evidence for Hebbian LTP, and outside of that, we can't really say for sure. Maybe they go a bit further. I do find that they tend to interpret that as, well, this is really evidence of certain synapses not being involved in learning. But yeah, they put a few caveats in there. I think it's not completely unfair.
And the number that they're getting, that's probably from D. Is that correct?
Let's see. Yeah, I think it is. Yes, because that would be the 50% in CA1, right?
Anyway, that's my question. I'm not sure if I understand.
Yeah, or nearly 50%, as they say. Good. Go ahead. Yes. Hello.
Oh,
Or should I?
yeah, I
Yeah.
just wanted to ask, do they not consider any kind of homeostasis happening with Hebbian plastic?
They mention it. So they mention this among the set of not learning related processes that have to take place for stability and balance, etc. They don't get into a lot of detail about it. But yeah, so they consider that needs to be present, but they're not incorporating, say, processes of homeostasis in their evidence for Hebbian LTP.
I see. Okay. Thank you.
All right, I'm totally happy to get into more questions because I know we don't have too much more left to go anyway. So anyway, where were we? Was I already on C? I'm not sure, but I think I was, right? So that's the evidence of the 50% low variance SASD pairs. And then in D, what they do is they're basically comparing three different things. They've got their variance and size, and then also the relative abundance, which is indicated by colors. So if something is blue, then it's under abundant or has low abundance, and red is highly abundant or overabundant. And you can see here that there is an abundant bimodality and also abundant low variance, but you can notice that in entorhinal layer entorhinal layer 3, there's less abundance of low variance large pairs, and the abundance of small synapse low variability pairs, especially in CA3, could imply that those are not randomly wired, but subject to Hebbian plasticity that has a small synaptic weight target state as well, or set point as well. So it might not be just purely coincidence that they happen to be going through that state of being small, but they may be aiming for it. And that's something we'll get into a bit more in just a moment. So after this analysis, in the next figure, figure 3, and I see we have another question. Go ahead, Ken.
All right, it's me again. Could you go back to D?
Absolutely. One sec. I just have to get my mouse to do the right thing.
So
yeah, here we go.
I just want to make 100% sure that I understand how they make this figure. So,
Yeah.
So what they did, correct me if I'm wrong. Okay. So the x-axis, what is it? It's the relative difference.
That's the size. Yeah.
And the y-axis is the mean. And so they would take all the same axon, same dendrite pairs and each pair would be a point in that space. And so
Yep.
they would plot point, 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 point. So that should be a scatter plot, essentially. And then they randomize and create another point, 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 point for the random, I guess. And then they smooth the heck out of that for a density plot. And when the densities are of the same-axon, same-dendrite is significantly greater than the random in that region of the space, they color
It was
red.
my okay. So maybe I'm not sure where you read that exactly, because it was my understanding that this isn't a density but a number, like the number of pairs that they found that are, say, for example, small with low variance.
But it's a 2D plot. So it's like the fundamental data is points. I'm also looking at the Mata paper. The Mata paper shows these same figures, but they also show points in another graph that is right next to it. You don't have the Mata handy in your...
I don't have it handy, no.
Okay.
Sorry. I should have perhaps. I mean, technically, I think it's possible for you to briefly, because I believe that Google allows us to have multiple presentations on at the same time. So if you want to pop it up, I think you can.
Oh, okay. I will give that a try. I don't have anything too bad here on my screen. Okay, so this is the Mata paper. You can see my screen?
There it is. Yeah, it just showed up.
Okay, so E and F here are, I think, the original way how they create these diagrams. And so there's this constellation of points for the same axon, same dendrite pairs, and that gets translated into this. And I think, you know, I mean, this plot of these heat maps, which is why I'm saying it's a density. When I'm saying it's a density, I'm saying it's the density of these points relative to the density of their randomly shuffled matches.
All right. Yeah, I was interpreting this as binning in a sense, but I guess density, that could work. Yeah.
Okay, binning or because they're drawing these black smooth curves here, and those black curves are what they're getting their percentages from, I believe. And this diagram here, which I swear to God, I've tried to understand this, but I feel like I'm not getting something. This is their argument about what in A there, what is consistent with Hebbian and not consistent with Hebbian. So in any case, I wanted to make sure that I understood this because it seems like what they're saying is that if you look in F here, if I draw these isocurves around the set of same axon, same dendrite matched synaptic pairs, and I look for those that are significantly greater than would be expected by chance, that those are the ones that are consistent with Hebbian plasticity. So those two red hot spots, and that is their fundamental number when they're saying 50% of the synapses in this region are consistent with, or they even phrase it as at most 50% is consistent with Hebbian plasticity, I believe.
Right. Yeah, and they do that the same, of course, in the other paper. Yeah. Okay, thank you. That's very helpful. Yeah.
Okay, I should have phrased that in the form of a question because I'm still confused. But go ahead.
Can you rephrase your question then?
Well, it really is. I want to make sure that first of all, I want to make sure that I understand, but also it's like the fundamental question is still, why is that particular cutoff what they deem to be consistent with Hebbian plasticity.
Ah, drat. Okay. I remember reading about the cutoff, and now I forgot how they said it and why they said it that way. If anyone here has read the paper and does remember, please do speak up, because I don't know the answer to Ken's question. Maybe we're just going to have to leave that for a later examination. Yeah. Okay. Then I guess we will head on to figure 3 now, and we can come back to figure 2 in the conversation at the end if we want to. All right, so here now, they're going to take a look at the sublayers of CA3 and CA1, and they start in CA3. They're going to take a look to see if there's a layer dependency of those distributions that they found. And in A, what they're showing you is that they have those subvolumes arranged in different layers. So for example, they have in stratum lacunosum moleculara, the input from entorhinal cortex layer 2, or at least that's what mainly goes in there. They have 2 volumes in there. I'm not reading it correctly. And in stratum radiatum, where you have the a lot of connections to CA1 onto the apical dendrites via Schaefer collaterals. They've got 3 volumes in there. And in stratum oriens, they've got another 3 layers there, or 3 subvolumes there, 4 subvolumes in there that contain mainly axons and inhibitory neurons. And the local connectivity occurs primarily via synapses that are located in the stratum radiatum and stratum oriens, while in CA3, the stratum lacunosum moleculara is expected to comprise mostly the long-range input axons from entorhinal cortex layer 2. Now in B, you're seeing a couple of examples of SASD pairs onto identified pyramidal cells in layer 3 CA3. And you can notice the large and similar on the left and small and similar on the right examples of these SAAD configurations. Now in C, all of the layers showed a strong deviation from the log normal synaptic size distribution, so they were typically bimodal. There was a pronounced overabundance of small synapses in SR and SO, but almost no overabundance in SLM when you consider the SASD paired synaptic configurations. Small dominated circuitry is therefore focused in the deeper CA3 layers where most of the local, so CA3 to CA3 synaptic connectivity is expected to be. There's an overabundance of low variance synaptic pairs, particularly in SR and SO. Then in D and E, where again they carry out these additional plots, they make these additional plots for the variance, and also here their 2D plot again. Evidence of Hebbian-consistent low variance SASD pairs appears in both the 1D and 2D plots. And you can see that there's a strong contribution of small synapses with low coefficients of variance, in particular in SR and SO, so in those deeper layers. It's abundant in both of those, if you take a look at those percentages there. So it appears like you have 2 set points in that local connectivity. And then taking a look at pretty much the same thing, this time in CA1, where they have 3 different subvolumes. There's 1 volume in the lacunosum moleculara, so where the input from entorhinal cortex layer 3 comes in. 1 in stratum radiatum, where you typically find the apical dendrites on Schaefer collaterals, which are the input from CA3, and 1 in the stratum oriens, which mainly contains, again, axons and inhibitory neurons. And the overall comparison by region had shown a substantial fraction of large synaptic weights with low variance large SASD pairs. So if we look at B, and this time they didn't give us examples of what they look like, but I assume it's pretty much the same as in CA3. Bimodal synaptic size distributions are primarily found in SL, stratum lacunosum of CA1, while long-range input from medial entorhinal cortex is thought to arrive in there. And you can notice again the percentage of pairs involved here. There's a shoulder of large synapses that's less pronounced in SO, the output of the hippocampus. And the most pronounced occurrence of large synapses was in SL, with these large synapses outnumbering the small ones in SASD pairs, which is a bias that wasn't found in any other of the studied cortical circuits, or even beyond this study, apparently, at least the authors say so. I didn't check if that was true. And then in C and D, there's evidence of Hebbian-consistent low variance SASD pairs, both in the 1 and 2D plots, and in particular for large synaptic pairs in stratum lacunosum. Notice those similarities in SR and SO between CA3 and CA1. So if we were to page back and forth here, and you just look at only SR and SO, you can see quite a bit of similarity there. That's interesting, at least in some sense, because to me, it says something about circuit purpose or function, I hope. Okay, so now that we've looked at these results, maybe we can do a little discussion of, or their discussion, look at their discussion of the various things they found in here. And we begin with the unique signatures. So they found some unique signatures. So in the medial entorhinal cortex, it shows synaptic weight distributions and Hebbian plasticity related signatures that are much in line with the reports that you find in other neocortical data. CA1 and CA3 show uniquely different synaptic weight distributions. Both of them share a population of substantially smaller synapses and a set of larger synapses, and they're matched in size between the two hippocampal areas. CA3 has an extreme overabundance of the small synapses that outnumber the larger ones by almost 9 to 1. And if you think of this in a model context, that might align well, for example, with a model of CA3 in a heavily recurrent excitatory network in which only some of the synapses are recruited into episodic or heterosociative attractors, which I can get into in a bit more later if anyone's interested in that aspect. CA1, in contrast, has almost 50% of synapses in large weight state. And the CA3 small synapses show signs of Hebbian low variance configuration in those SASD pairs. And that suggests that those are not random, even for that small synapse pool. So they could be used as information storage in small synapses, in other words, intended to be small. So again, if we think of this in modeling terms, that might be because there's an important distinction there between whatever the large synapses and the small synapses are indicating. And in some contexts, in models of sequential storage of heterosociative connectivity, you could make a distinction there between autoassociative and heterosociative patterns, which I think I get into a bit more at some point here. So even in the CA1, there are only up to about 50% that reach that significance that Ken was just talking about for pairs that show significantly reduced weight variance consistent with saturated LTP. And according to authors, that challenges a view where all of CA1 is involved in information storage, or all of the synapses of CA1 are involved in information storage. So there are parts of the circuit that wouldn't be. But, if you think about this in terms of modeling, then again, that somewhat aligns with the need for things like recurrent inhibition, synchronization of sparse patterns, these sorts of things that wouldn't be part of the actual learning, but would still be required for the system to operate correctly when you're doing acquisition and retrieval. Now, the sublayers of CA1 and CA3 have unique properties, with SR and SO being primarily the sites of possible Hebbian plasticity via the small synapses in CA3. That's consistent with the needs that I was just talking about for this broad auto and heterosociative coding, if you're looking at models. But SL is dominating in large synapse pair consistency in CA1, and that would seem to indicate a strong influence of entorhinal cortex layer 3 input on CA1, which, I don't know, again, if we think about this in terms of models, what could that mean? Could align with models where entorhinal cortex layer 3 entrains sparse encoded patterns with a certain meaning with an association with context. in CA1. It's one possibility. I'm just putting that out there. All right, so now about coding. The bimodal distributions that are seen in the hippocampus don't appear in entorhinal cortex. The small synapse low variance population seen in the hippocampus wasn't seen in the medial entorhinal cortex or in many other cortical regions. And they do not appear to be random synapses in waiting, or at least that's what the authors are saying. But they may themselves serve information storage in these consistent high multiplicity connections from two or more. Let's see who's got. Oh, it's Ken again. Hey.
Sorry.
No, don't say sorry.
I just want to unpack this, make sure I understand. So the assumption that they're making is that if two synapses are large and they're large together, then that could be Hebbian. But if two synapses are small, then that wouldn't be Hebbian.
This is not what they're saying.
That's not what they're saying. Okay.
Nope. No. They're saying exactly the opposite. They're saying that if two synapses on the same axons and dendrites are small and they have low variance, so they're similar, then again, this is pointing to a similar history of pre and post synaptic firing. So it's consistent with Hebbian LTP, but it's Hebbian LTP aiming at a different set point. That's at least what they're claiming. Now, it doesn't have to be. Later on in another slide, we're going to mention that this could just be a transition, right, from small to large or large to small. But it's interesting that there are these two peaks. So it's not as if you see this graded sort of, there are lots of different sizes and always consistently low variance. So this doesn't look like a transition. It looks like there are two different set points.
Because the standard model, I have thought that the standard model is kind of, and this is what they mean by like a training synapse or population in waiting. Okay, it's like, I've got a dendritic spine that goes out and reaches out at random, and it's a silent synapse that's listening for coincidence, and if it doesn't hear any, then it shrinks back, and if it does hear coincidence, then it expands and stabilizes. And if I had random ones of those going off at different times, then if I had the same dendrite but a different spine that reached out and happened to hit the same axon, different place, then it would also get coincidences and that would grow. Yep. And so what you would expect is under that scenario, there would be a population of matched large, but there wouldn't be a population of matched small.
Right. That's the default. Yeah, that's the default model.
And the fact that they're seeing a bunch of matched small is really against that standard
That's
model.
what they're saying. Yep.
Very cool. Okay.
But they're not being absolutist about it. Of course, they put caveats in there and say that if they are indeed synapses in waiting, then you might still expect to see a large population of these synapses in waiting, staying relatively small, and so it could be a transition phenomenon.
Have any of these papers done like simulations of this in the sense of kind of
well, they didn't.
Simulations that...
Oh, that's a good question. No, I don't know. Maybe. Yeah. Sorry. I thought you were aiming at something else because you said simulation, and my mind immediately went to my next point, which is that having these two different set points, if they are two different set points, it does actually make some sense from a modeling point of view. Because if you imagine what you would need if you have and I'm saying this because those were some of the models that we worked with back in Boston University and stuff. If you have a CA3 model that is useful both for the autoassociative patterns that are already present in entorhinal cortex, but that also then within CA3 encode heteroassociative connections, so across a sequence. You can imagine if you had this smaller set point how that would help differentiate it from autoassociative, but still make possible the retrieval of episodic memory only when there is a queue of multiple patterns over time with overlapping EPSPs. You can imagine how that might work or might help if you have basically a weight matrix like that, where the diagonal is strong and you've got one of the triangles filled up with this lower set point. I'm not saying that that's how it is, but you can imagine how that could work. Just as a, a thought experiment. Moving on to the next slide here, which is about their discussion of Hebbian learning. So this is kind of just pulling a few things together from previous information. So Hebbian learning traces from these paired synaptic configurations. The use of these paired synaptic configurations for the analysis of properties of Hebbian learning actually has quite a bit of history. It was pioneered by Sanofsky and Harris and others. Bartol and Bromer et al. In 2015, they used that to estimate storage capacity or bit depth per synapse. So in that paper, which was a nano-connectomic upper bound on the variability of synaptic plasticity, they established that single axons make 2 or more synaptic contacts onto the same dendrites, and those tend to have shared histories of presynaptic and postsynaptic activity. Now, we've already talked about that previously because we had a few questions about it. In Mata Burning et al., 2019, they applied this large-scale 3D EM and used it to determine which fraction of a synaptic circuit was consistent with a history of saturated Hebbian LTP, so using pretty much the same approach we're seeing here. And in primary somatosensory cortex, only about 20% of the synapses could be considered Hebbian by these standards. Now, even in CA1, if you're using this method, only up to about 50% of the paired synaptic configurations are consistent with that, with a history of saturated LTP. So perhaps even in a circuit that's thought to be dedicated to information storage, I mean, hippocampus is so essential, so important to that. A substantial fraction of the synapses are needed for non-storage related purposes. For example, regulating excitatory inhibitory balance, maintaining stability, things like that. I mean, for instance, you'll want to have interneurons connected to silence activity after every run of action potentials and to synchronize. So this would be a typical type of use of those synapses. Now, there's a word about this being an unstimulated study. They looked at the disadvantages and advantages, or at least they mentioned them, because as mentioned, these are mouse brains that were analyzed at 4 weeks of age with a history that you would typically find in a lab mouse. And you'd imagine that the disadvantage is that these are pretty monotonous lives. Those mice don't get a lot of exciting existential stimulations that they have to figure out about their world as you would in the wild. And you could imagine that if you give them something like enhanced stimulation, at least compared to laboratory conditions, for a more enriched environment or trials that you're carrying out, then you could find increased plasticity, consistent synaptic properties, and you might find even more pronounced synaptic weight distributions. But an advantage of this approach, or this particular study, is that it's studying the brain's memory circuits in a native state, that those circuits have had to store relevant information at what you might consider a normal rate without any specific trained skills. And so it gives you a baseline, and that baseline already shows clear and succinct features of synaptic weight distributions. As for key sites of learning, these were determined by the sublayer-specific analysis of the synaptic configurations. In CA3, SR and SO stood out for possible storage in small synapse configurations via Hebbian plasticity, and the stratum lacunosum in CA1 for strong overabundance of large synaptic pairs with low variance. That suggests that you can focus on these key synaptic connections in future studies that are involved in information storage. And those studies then could be aimed at uncovering synaptic changes in response to novel stimuli. So this is their proposal for taking this on to the next stage in a way. Okay, so graded versus bimodal weights. The results here showed strongly bimodal synaptic weight distributions in hippocampus, particularly in CA1. Most of the literature and models, they say, assume LTP leads to synapses reaching graded synaptic weight set points. And then, of course, the synaptic storage capacity depends on the number of those differentiable set points. The recent analyses, like the Dorkenwald paper, revived the notion of binary synaptic storage, where a synapse is either in a learned state or not a learned state. And this is, of course, the publication that we presented here back in July, where they studied the evidence for binary modes in mouse primary visual cortex. One could interpret the bimodal distributions like that. We could just stick with that. But the results that they have, at least the ones in CA3 for the small synaptic weights, might be the result of plasticity as well, in which case you then have at least two learned states, or a trinary situation. All right, so a little bit about network models. Synaptic configurations are not consistent with saturated LTP versus the synaptic configurations that are not consistent with saturated LTP versus not saturated could be interpreted as being in the process of forming such storage. So they acknowledge here that this isn't cut and dry evidence for multiple states, multiple set points. They could be in that process of formation or in the course of dissolving previously stored information. And you can imagine that that's particularly likely in the hippocampus, because that's assumed to play only a intermediate term role in information storage. So you would have substantial turnover of synaptic weights. But they suggest that to further explore this in terms of network models, what you should do is you should fit kinetic models of synaptic plasticity and turnover to the data. And then you can come up with more detailed plasticity models. You can refine them a bit more and test them. And I think that would actually be very interesting. So this was data that wasn't available way back when I was still modeling hippocampus. And I think I would approach that a bit differently now, given what we've seen in Dorkenwald as well as here. Now, I wanted to put this, before we end, I want to put it in the context of memory decoding. And I'm very interested to see also what Ken and Ariel and others think about this. So I think that this is a nice example in the sense that it uses basic static volume EM data. There's really nothing else required for this. There was no specific memory identified or decoded. So in that sense, it's not a memory decoding paper, obviously. The experimental setup wasn't meant to do that. But if you are going to do memory decoding from EM data, for instance, then what you need an understanding of which synapses in a circuit store learned engrams and which one have supporting roles. You need to understand the meaningful coding. So is it a gradual coding, or are there set points, and how many set points are there? And then, of course, you can benefit from an understanding of the implications of different coding in different regions or sublayers in terms of encoding and decoding operations in the circuit. So as a source of information about the sorts of things that you should care about and that would be relevant if you're trying to do memory decoding from a static set of brain data, I found that this was actually very interesting. It's not the only paper. There are others, and we should put them all together and just kind of collect it all, I'd say, in some giant map of things to care about. But from that point of view, I found that this paper was quite interesting. And that was my last slide, I believe. So thank you very much. Let's talk about it. Go ahead.
Thanks, Randal. It was great. So we've got these three papers: the Bartol paper, this paper, and the Dorkenwald paper. My assumption is that the Bartol paper is incompatible and should be interpreted as getting very, very lucky with a small number of matched synapses, because they basically were saying, hey, these are so matched that we can essentially quantify the noise as being this fraction of a synapse.
Would you call their data cherry picked?
It's not cherry picked because the size was, I think, something like less than 10 by 10 by 10 microns. So, which brings up one other thing that the Dorkenwald paper did, but I'm not sure if this paper did. The Dorkenwald paper split between synapses that were very far apart, matched synapses that happen to be far apart versus close together on the same branch of the dendrite, let's say. Did this paper do that?
I didn't see anything about that in the methods or anywhere else, so I don't think so.
It's a bioarchive, so they might add that analysis,
Right.
I suppose. In any case, the other thing I wanted to say is that this paper, my understanding, this is a question, is that this paper and the Dorkenwald paper, I know they're in different regions of the brain, okay? But the Mata paper is in a piece of somatosensory cortex that could be compared to visual cortex. They are not incompatible. But the analysis is incompatible because this is essentially saying, hey, Hebbian plasticity will move toward a saturation point that is the same for all synapses. And the Dorkenwald paper is saying there's literally a binary bam, bam, small synapse, large synapse, and there's an analog variable of noise on top of that. And that analog variable, assuming that model, that analog variable, depending upon how big it was, could completely swamp the statistics of this. And you could have every single binary variable tracking perfectly, but if the analog variable was big enough, then you would, quote unquote, by this analysis, rule out them as compatible with Hebbian plasticity. Is that your?
But can I ask? So you said that they're incompatible because Dorkenwald says there are these two different
Well, I'm saying that they are compatible. That the data is compatible, but not the analysis, but the
interpretation.
The interpretation. But the data is compatible. I think that that if you were to take this data and put it through the analysis of the Dorkenwald paper, that you would get the same type of thing, and vice versa.
I think that's likely, and I would have kind of liked to see and preferred to see the Dorkenwald analysis applied here. It would have been pretty cool.
Yeah, that would have been cool.
Yeah.
But the difference is that there's this analog variable that is assumed to be complete noise, and this paper doesn't assume that, I guess.
Yeah, no, didn't add it in explicitly anyway, and I don't think they mention it either. So no, I wonder. Yeah, I mean, obviously they were very aware of the Dorkenwald paper, so I wonder why. Yeah, good question. Go ahead, Andreas.
Yeah, thank you for the explaining this. And I'm curious, comparing CA1 and CA3, those two graphs, and I guess with the understanding that CA3 goes into CA1, why, and if you mentioned those two set points, so why would CA1 have, a more of the set point than CA3? Why? What's happening? How do you interpret that?
Sorry, more of the set points?
Well, you have that bump and it goes higher. So I think that there's the way, if I understood, there's these two plateaus. There's the top and there's the shoulder. And in CA1, the shoulder is higher. And so that means it's it's representing a more frequent, a greater frequency relative. And so why is CA1 different than CA3 in shape? How do you interpret that?
Well, these are relative frequency though that we're seeing here. So I'm not exactly sure how to read that, because the only thing they mention in the paper is that the number or the frequency, the abundance of the small low variance SASDs is much greater in CA3. They don't necessarily say that if you look at them as an absolute number, that you can compare them that way. So I'm not exactly sure. I don't know if anyone else has found it in the paper. Yeah.
I mean, so if I understand the graph correctly,
Because you could either look at it, right? You could look at it as in CA1, for some reason, there are many, many large and many, many small. Or you could look at it in CA3, you've got way more small for some reason. And they mention both of these in their results because they point out the prevalence of small in CA3 and that there are many large SASDs in particularly the one of the layers of CA1. I'm not sure how to compare that directly.
well, and so it just seems like a divergence, that there's a divergence being coded, and and that seems fundamental. I think it's very interesting, so I mean, just to be able to take things from a one-letter alphabet to a two-letter alphabet, let's say, right?
Yeah. Yeah.
And when I think of entorhinal cortex, and I'm just a newbie, but I think of it, there's what and when, I mean, what and where, let's say, is matched together. But I think of it as what and how, so you have like sensory information, and then you have what I would call, I don't know, process information. And so if those are coded, let's say those are coded distinctly, those are two different things. I don't know, but that would be very meaningful. I'm just babbling, but thank you for your talk.
Yeah. Okay. Anyways, yeah, it would be interesting to look at the difference of say entorhinal layer 2 and entorhinal layer 3 and the information they provide into the hippocampus, and therefore what you would expect to find in CA3 and CA1 in terms of how it has to influence the coding of patterns engrams there, especially since we're talking about sparse engrams in hippocampus. Because, for example, in entorhinal layer 2, so you mentioned the what and how, or was it the where? I'm sorry, I'm forgetting. But it wasn't the when, right? The important part is it wasn't the when. Whereas when I think of entorhinal layer 2, I think very much of the when. I think of a lot of a buffering of temporal data, repeated activity, things like that coming in from entorhinal layer 2. I'm not sure if you agree.
Well, if I could just state my point, in the little that I know, but if you have, let's say, a rat traveling and it's storing information about where it's been or where it's going to be compared to what it will sense. So there's this what information and this where information. But when I hear about that, just metaphysically as a philosopher, like the levels of knowledge would be whether, what, how, why. And so it's not really like what and how is a very natural combination, you see. And so it may look like where, but from
this makes sense to me from
it's
EC3.
more like how. That would be my thesis. And
that makes sense to me. And when I was thinking of how this might fit into models of hippocampus, that's also kind of where my mind was going with those strong connections in CA1 from entorhinal layer 3, was that, yes, this is providing you context from something that is a more distributed code of what has actually happened, which gives you the what and how and those sorts of information, which puts it in that context. And that's important for CA1 because CA1 is leading to the output, has to take that information back out to be understood, to be meaningful to the rest of cortex again. Whereas
it's
in
a disentangling, a metaphysical.
It's a disentangling. It's a recoding in a sense. Yeah. From the sparse coding in hippocampus back out to the more distributed coding that you find in cortex.
And
of
that
course,
can be
that
part
happens
of
in subiculum as well.
If you can do that, you can compress. I think that's.
Well, you're doing a lot of compression through the hippocampus. Yeah. Like it's almost like you're giving it labels. Yeah. And then or at least I should mention quickly, caveat, these are all just model ideas. None of this is like proven. But anyway, so yes, labels there and compression, because labels do compress. It's like vector coding. And then in CA3, where you're getting the input from entorhinal layer 2, the interesting thing there is CA3 is heavily involved with the episodic coding, with the creation of these recurrent connections that encode heterosociative dependencies between different patterns, and that matches up with entorhinal layer 2 providing a lot of time, not necessarily time in terms of intervals, but order information, sequence order information from patterns. So this is a different kind of information. That's more of a when, but not really a when with a particular timestamp, but more of an order of this, then that, then that, then that, coming in from a buffer that's repeating that kind of activity. And that's leading into CA3. And if you want to encode something that is a sequence and you want it to reactivate, to retrieve only when you give it a cue that is not just, in an autoassociative cue, you give a piece of the pattern. In a heterosociative cue, you give pieces of patterns, but of a sequence of patterns. So you might give like the first 2 out of 6. And that's exactly what you would expect to find then, is that you would need to have connections, recurrent connections that are not quite strong enough to cause retrieval upon receiving just the first sequence, but that would achieve retrieval upon receiving maybe 2 or 3 out of the sequence with overlapping EPSPs. So that's why in my mind, that kind of clicked, that having these set point at a lower synaptic strength would be meaningful and useful in CA3. Of course, saying that from a model point of view that would be useful isn't evidence that that's actually what's going on. So I don't know. But that's how I was interpreting this as, oh, this could help. This could be useful at least. Yeah. Are we already over time? I can't see a clock from where I'm sitting, so I have no idea where we are.
Yeah, we're definitely over time.
Oh, oops. Okay. All right. Well, are there any more questions? Because I don't need to rush away, but also feel free to leave if you've heard enough. And Ken, I don't know if you had any input to my whole very complicated and messy and probably confusing spiel about models there.
I think it's I would love to discuss hippocampal models with you and hetero associativity and homoassociativity and stuff like that at some.
Just keep in mind they're just models. They're just hypotheses, right?
Yeah. But that would be great at some time.
Yeah, sure. Okay. Well, then I guess all that remains is to say thank you very much. It was a pleasure to present this one. See you all later or next time. And maybe at SFN if you're going there. See you there.
Thanks.
Right. Bye-bye.